How recording? Bismillahirrahmanirrahim. Good evening, everybody. I welcome all of you on behalf of Science International in this premier one and only unique session on randomized control trial. It's an orientation session by the famous Dr. Mujtaba Qadri, who has a tremendous shining medical practice record as well as his mentorship during his past decades. It's spread over decades, not a matter of 1 or 2 years. He has been. I'll cut it short. I don't know. You can see the detailed introduction uh, in the promo and posters on the website. But here I just take a few minutes to introduce him as he has been, as I know him since I think 2010-11, while we are working in Saudi Arabia. And I know he has been the chairman of medicine at Shifa International Hospital, I think chairman of nephrology, and then he has been the chairman of medicine at King Abdulaziz Medical City under National Guard Health Affairs for quite some years. He has been working with, I think, in Riyadh, and then he moved back to Pakistan. He worked with National University of Medical Sciences. I think he has been chairman and director for research with NAMS again for many years. And he has trained, thousands of students. I think more than 3,000 students has been trained on clinical research by Dr. Mujtaba Qadri. So we are honored, Dr. Mujtaba, to have you with us, and our students are looking forward towards this very insightful talk. uh, I hand over the rostrum to you. Please go ahead, Dr. Mujtaba.
Ji, Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh, everyone. And Jazakallah khair, Ata Saab, for a very generous introduction. It's just that if you are there in the field for such a long time and in the seventh decade of life, you accumulate some experiences. That's all I have, nothing more than that. But I'm happy to share whatever has come my way as a further trust that has to be spread as much as we can, inshallah. So today I'll just give you a little bit of an orientation to clinical research and try and build up a little bit of excitement and interest, a curiosity, so that people get into research and understand that without research, there's no innovation. Without innovation, there's no entrepreneurship. And this is in this 21st century, I think most of my audience and others that I deal with beyond Generation Z, I think आजकल कुछ alpha, beta, theta पता नहीं क्या आ गया है. So all of you are entrepreneurs. Not everybody, is just going to take office jobs and all that. You'll all be innovators. And for innovators and in this artificial intelligence era, clinical research and its translational aspects are also an exciting career option for people from any background. So one can be a physician, one can be a nurse, a pharmacist, nutritionist, physical therapist, and social worker, and behavioral scientist, psychologist, and one can do clinical research for the reasons we will see. And I will try and give you an example from an experience that I shared with Atasab out in Jeddah National Guard Hospital some years back, and tell you how that particular project arose and what were some of the findings from that particular project as we go along. So, just to move right ahead, I'm very, very grateful to Science International for giving us this platform to be able to share this Ilman Nafian. Hopefully, that will be a two-way street. I request people to write any questions they have in the chat so that we can try and address all of them, inshallah, in the end. So my brief background, I mean, like it's just a very long story, and all I can say is that I've spent some time on Earth, so that's why one can see a few appointments here and there, which all of us go through, and which are not the end for anyone. It's just that everyone has a different experience. And with that experience comes the responsibility of sharing that experience. So, just to give you a little bit of an overview of clinical research, what is it? It's a medical research process that studies uh, people. So, it's a study of human beings. So that is the difference between all other forms of research. That's a study of human beings, of people, or their data and samples. And it is mainly done to understand and improve human health and its outcomes. And it can focus on developing new ways to detect, diagnose, treat, or prevent disease, and can have multiple different types of studies, including clinical trials, which we'll give you an example of. And it is something that is important in the way in the route towards development of new medical treatments, medications, devices, nutritional products. All those things come in as a result of somebody doing some form of clinical research where it involves human beings at some stage, either as volunteers or as patients. The purpose is to develop new treatments, find and test new drugs, therapies, medical devices, and also to improve current treatments. So, खूब से खूब तर की तलाश में, we want to, develop newer ways of therapy or newer management strategies to improve outcomes, of course, which is the ultimate goal. And it can be to prevent disease, to detect disease in the form of diagnostic tests. New tools can also come in, and to also understand disease causes and progression of disease as has a natural outcome, so to speak. But the ultimate aim is to improve health services, cost effectiveness, healthcare outcomes, and uh, the main thing, unlike lab research, is that clinical research, like I said, it involves real human beings. So either by interacting with them or by taking their samples or their data, and all of them require the integrity of good clinical practice, which is a required certification that people need to go through by the International Conference for Harmonization Good Clinical Practice agreement that came about in 1996. And if anyone is involved in clinical research actively, one needs to be certified by this ICH-GCP. There are various courses online. One can do that, and they remain valid for 2 to 3 years before something new comes in, and we have to keep exploring that. A lot of those the goals of clinical research are safety of a new medication or the dosing and or device or new forms of treatment, and also to measure effectiveness or efficacy as the case may be. A lot of human volunteers are putting their lives on the line to help advance science, to help us use medications which come out in the market almost every day. But for one single medication to come to the market, there's a huge story, and a lot of people, it's like a marathon race. A lot of people don't complete the marathon, and they drop out at the beginning, in the middle, almost in the end also sometimes, which is a big cost that industry and society has to pay. So if one does good clinical research, valid designs, then studies are generalizable and they have that internal and external validity in order to be trusted as studies before a new drug is, brought into the market. And it is the process of this research is highly regulated, and a lot of this involves strict protocols and criteria to ensure mainly the safety of the participants and that the results are valid, outcomes so that can be used by society to improve healthcare. So one can have clinical trials as one of those forms where you test new interventions in people to determine safety, efficacy in various forms. Or one can simply have clinical research where a natural experiment or natural exposure of nature is taking place, and we are simply not intervening, but just seeing the exposure and the outcomes in the form of observational studies, and different types are available to us. And we will go through some of these and some minor details. There are natural history studies also, which also looked at health information collected from different sources or registries to see how disease develops in time. But then there are behavioral research. So one could have behavioral research, psychological interventions, or some form of dialectical behavioral therapy, for instance, or other forms of interventions. These are all there, and also require the same integrity of the research process. Or one could have health services research, which looks at comparative effectiveness of research, whether it is different forms of policies or procedures to see the health outcomes of a community or society. So, if you want to just classify these clinical studies in the simplest form, excuse me, one can just talk about descriptive studies, and a lot of descriptive studies, at least in our part of the world, in Pakistan, a lot of descriptive studies are done, and some people do it all their lives. And then they also have their value, what happened to whom and where and how many, and what was the percentage of people being, who were affected by this, or what was the percentage who were not affected by this, and what was the gender and what was the age group, and these things which are generally descriptive in nature, just by the word descriptive. But then, if you want to stimulate your brain towards more translational research, and then one needs to get into analytical studies. Analytical, by the name, the way it suggests, it has a little bit of an intellectual, aspect to it, which makes us think, which makes us analyze and look at things critically. So even in there, those non-experimental studies where you don't intervene yourself, but have a natural exposure and outcome, or you have an outcome and you see what was the exposure in those who had a case or versus those who had no case, or a single point in time, so-called prevalence studies, cross-sectional studies. These also have some bearing, but they can only show what are known as association. They do not establish causation. For us to determine causation of something, we have to do an experiment where, as investigators, we intervene. And in those experiments, some experiments are done without any element of chance, and you are selecting cases and controls in the form of exposure and controls, or experimental group and control groups, who can be either concurrent or can be historical controls, but in a non-randomized manner. Or one can randomize by chance alone. The people could be assigned to a treatment intervention, or a diagnostic intervention, or a behavioral intervention, or a device related intervention, versus no intervention, or intervention against matching substance which looks, tastes, smells, weighs the same, called a placebo, usually does not have any active principle. Or it can be against an active control, where you have a standard of care and you're comparing things directly head to head with a standard of care. But when you do randomization by chance, you can, like I said, so you can have the control, which is a placebo, no treatment, or you can have some different forms of doses that you see the response against, or you can have an active therapy that you're comparing head to head directly with, for either superiority, that you are superior to that particular therapy, you have to show some margin of superiority, or you are equivalent to that particular therapy, or at least you're not below an inferiority margin, that the so-called non-inferiority, that you're almost 19-20, or 19-20. For those who don't speak Urdu, understand that. So in that range, but it is not below a certain margin. So it is almost as good as the original, but maybe slightly inferior effect, but at least may have less costs, may have less side effects. And so these are also designs that uh, pharmaceutical industry uses to bring in new drugs into the community, which are so-called Me Too drugs. So this is a typical classification of clinical studies, and just to give a quick summary of that. So when you're looking at a cross-sectional study, you take a population and you take a representative sample of that population. And then at the same point in time, you see people who have a disease and and are exposed to a certain intervention, and you see people who have the disease and are not exposed to that intervention. And you also see at the same time those who do not have the disease but were exposed to the intervention, and compare them to those who do not have the disease and were not subject to an intervention of nature or exposure, whatever you want to call it. The only problem with these is that you can get so-called odds ratios from these as well, but you don't know whether the chicken came first or the egg came first, because these are point prevalence studies which are done at the same point in time and can be hypothesis generating. Okay, can I just request everyone to mute themselves, or maybe let me just try and see if I can do that from my side. Just a sec. If I can do that, that'll be great. Where is this option to mute? Um, so if I can request everyone to mute themselves. Okay, I can hear people's voices in the background. So just mute yourself. Just click that audio so that at least you're muted. Thank you very much, because I can't locate it right now. Okay. So going back to these designs, the second design in the observational designs, that is probably something that can be done relatively, easily, but also requires some specific methodological validity, is to take rare diseases and see if if you have a disease and go back in time whether they had an exposure of interest or not. And similarly, you take a control who are typically almost like identical twins of the patients or those with disease, except that they don't have disease. And then you also see whether they received an exposure or not back in time. So just by the nature of this direction of inquiry going retrospective, there may be missing data and some various forms of bias that we can study on another occasion, inshallah. But then the better form of observational designs are the cohort studies, in which you can have prospective cohort or retrospective cohort, where again you take a population and study patients without disease and see if they are exposed to a particular natural event or natural exposure, and in point in time, do they develop disease or do not develop the disease, and then also compare them to those who do not who are not exposed to that particular factor of interest, and yet they also either develop the disease or don't develop the disease. And then again, if you compare the two, these are known as incidence studies, and you can get a relative risk. So if you were to rate these three, this would be at the bottom, the cross-sectional one. Good to do some initial hypothesis generating work and population level prevalence studies. Then case control may come just above that in terms of level of evidence, and cohort would be pretty good, except that again, it is below what are known as randomized control trials, which we will see an example of, inshallah, today. And you can, but even in all of these, you cannot establish causation. But here, relative risk, here odds ratios, and here odds ratios, if they have very, very high values, like the effect size of the odds ratios or relative risk, if it is very high, 4, 8, 9, 10, as opposed to 1.3, zero point 0.7, levels like that, then you can also assume that maybe some form of causation is occurring. So, with that, I move to what is known as the translational continuum. So, in general, whenever we see something affecting a population, there's a huge story of 20 plus years that goes into it at the back, which happens in the labs or happens in animal studies or different forms of bioinformatics tools. And then before it comes to the bedside in the form of volunteers and safety and tolerance and efficacy studies, and then they go on to patients in the clinic, and then have some community based or population based effect. And this is a two-way street. So each time something is postulated out here, not always it benefits community or population, but then you can always go back and see, can we tweak that, the basic science a bit here, and see if the results out or next time around would be better for the community. So in general, whenever you have uh, something that comes out at the population level where there's true benefit to society, uh, then you need to understand that, for instance, if a drug comes into the market, which is an antibiotic that you may get from your store, initially that antibiotic may have had several family members who died in the process, that antibiotics, family members, the so called molecules. So, initially, when some molecules are extracted from plants or fungi or different sources, then they are purified, they're characterized. Somebody says, okay, this is their structure. Then somebody studies them in silico models and bioinformatics tools where they look at the structure of those molecules and see if you postulate that if you were to bind them to a receptor on a certain cell surface, for instance, and internalize it, what kind of effects may occur. So these are simulations, AI simulations. There's a simplest way of understanding that. And when things like that start happening, then you do cell culture studies where you take immortalized cells which are, grown outside in different circumstances and proliferate. And those cells serve like, living cells, but outside the human body. And those all outside the animal body. And in those cell cultures, also, you study those effects. Once things are postulated, yes, things may work. Then you go on to animal studies, small animals, mice to rabbits, some forms of fish. And then once those studies are done, then you may go on to larger animals like doves, and then it can go on to primates like monkeys or chimpanzees or apes. And all of these studies in animals have to be done with a lot of humane, form of interference. You can't give pain. You can't have more than the number required to do a study, do just extra animals and extra sacrifice of animals. You have to minimize the pain, minimize the number needed. So reduction and replacement and refinement are the rules you use in animal models. Then once you find out efficacy or safety or toxic or lethal dose, you use the safe dose from those animal studies, only 1/10 of that safe dose in human volunteers, maybe one, two, three, and see if they develop any side effects or not, and can they tolerate the dose or not. And you can escalate the dose a little bit. You can see if out of three people nobody develops a side effect, maybe take another three human volunteers. So these are people who are still taking risks for you and I, so that science and treatment and humanity can benefit. And then once you go on from 3 6, but if you have about 2 out of 3 people having side effects, the first 3, that's the end of that drug. That's the end of that molecule. And you may lose 4 or 5 years of work right there. But then if people can go through that to 3, 6, 20, 25, 25 to 100 volunteers, the so-called phase one studies where you look at safety and dose tolerability. Then you go up to phase two studies where you take between 100 to 1,000 or so volunteers and study the ongoing safety aspects, but also whether there's some efficacy of that drug for what you postulated in a particular system. And then once you're done with that, then you go through phase three randomized controlled trials where you take patients and patient population in which you test the drug against the control or placebo, as the case may be. And then if you have superiority in at least two of those studies, large multi-center, mostly clinical trials, then the drug is approved by a regulatory body, like for instance the FDA or the European Medical, EMEA or Pakistani DRAP. All of them would use some kind of studies to determine whether things need to be approved. Then once they're approved, the drug comes into the market and you have about 10 years of post-marketing surveillance. And in those phase four clinical trials, then you may drug may still develop some abnormal reaction or adverse event or adverse reaction or adverse drug reaction. And these can be idiosyncratic where they're unexpected and allergic reactions or other things which were never predicted, and the drug is out. It can be withdrawn from the market. And again, you lose 15 to 20 years of work there. So eventually, only about 20% of the evidence generated here unfortunately translates into the community for large population state effects. But that is generally a rule for evidence-based health practice. But most of these drugs which then make it, the few that make it, then start getting used, and then you see whether they need to be replaced or they have too many side effects or what one needs to do to come up with new drugs. So this is the way the system works for most randomized controlled trials. This is the simplest of the strategies for randomized controlled trials where you take a population of interest. So you have a target population and you take an accessible population which shows up in your clinic or hospitals or areas that you're studying at. And then from that, you take a sample or a representative sample and expose them to an experiment, which is in this case a drug or a new device or a new form of behavioral therapy, whatever it is. And by chance, some of them, about half of them, will not be exposed to that drug, maybe only get placebo. And then you see who develops the outcome, predefined outcomes, primary outcome, or secondary outcomes of interest. And again, in both the groups, with the exposure and without the exposure. And then you again can compare relative risk or so-called hazard ratios, and then you report them in different forms. This is the simplest form of randomized control trial, and there are several variants of that, which if any one of you attends a future course, inshallah, on randomized control trials, all those things are there in a lot of detail. So coming back to these studies, so we looked at some descriptive studies, maybe in the form of it can be case reports, single case, unusual case, and people describe how they presented, what were the features, what were the outcomes, what was the management, to a series of cases put together as case series. And but what we talk about on in hospitals as nutritionists or pharmacists or physicians or nurses or physical therapists, if we are simply using our expertise in there, we are giving an expert opinion based on some background information. Let me just answer this call from the hospital. Just a sec, please. हेलो अस्सलाम वालेकुम जी जी अस्सलाम वालेकुम जी जी जी
अली डॉक्टर मुख्तार के पेशेंट है इनकी कल राइट की डिप्राइड में हुई क्योंकि डायबिटिक फुट की पेशेंट है तो सर
बेटे इनका ना अभी जो है ना I'm doing a session online, तो can you just call Dr. Humayun, he's on call, ठीक है? Thank you so much. हम बस वो फिर ना सेकंड ऑन कॉल तो मैं ही हूँ, लेकिन जब मुझे गिव मी हाफ एन आवर, ठीक है? थैंक यू. ओके, सो from this particular thing, all I wanted to tell you was that what we talk about on rounds are just expert opinion. But if we just use any of this evidence, then case control studies that we just looked at in the previous slides, or would be somewhere here, cross sectional, to shayad may not even make it to this evidence. उससे ऊपर cohort studies, but अगर cohort studies भी अच्छे किए जाए तो they can almost match randomized control trials जो उसके ऊपर आ जाते हैं. But randomized control trials could be the best form of a clinical research original study. But उसको जब आप combine करते हैं उस evidence को, तो वो synopsis of synthesis, summaries से लेके बात all the way systems तक जाती है. Those of you who have a medical background will know that all these things exist in practice: evidence-based medicine, nursing, ACP Journal Club, up-to-date guidelines, Cochrane reviews, and all these things. But eventually, artificial intelligence ke saath, if things are then incorporated at the point of care, computerized, feedback and interactions and advice and alerts, then that systems, that is evidence at the point of care through the form of electronic health records, which incorporates clinical decision system. So clinical research can take any of these forms. One can combine other people's research and do systematic reviews and meta-analysis, and that's great. But when you do these things, original studies, then they have their own integrity, their own value. And maybe in the next 3-4 weeks, inshallah, we will be doing a 3-part series on randomized controlled trials, which is the highest form of original clinical research, I would say. So just to now move forward, in any time we come up with a research question, we have to keep this mnemonic finer in mind. Okay, is that particular study even feasible? Is it interesting enough? Is it novel? Is it ethical? Is it relevant to our practice? So when you come up with that, the feasibility में resources आ जाते हैं कि whether you have enough time, you have enough staff, you have enough funding, grants availability or not, and is the study design appropriate and feasible? And would you be able to get an adequate sample size? And do you have the trained staff to do that? And this is something that happened during the COVID era in Pakistan. A lot of people started doing clinical trials and they had no idea how to do clinical trials. But there was lots of money coming in, lots of pharmaceutical industry sponsors were pouring in grants, and sponsors were coming into Pakistan during the COVID time as they were in other parts of the world. And people were just jumping in and learning on the job, so to speak. But is their question interesting enough to investigators, to the peers in the community and the community at large? Is the question filling in a knowledge gap? So what if we do this study? So what if we do not do this study? How will it help in advancing practice or management or science as a result of this, answering this question? And most importantly, is it ethical? Is it ethical in the terms of who are we studying? Are we protecting vulnerable subjects? Are we, fairly including people, not unfairly excluding people? Is the study of such integrity that it is valid? All the inferences are valid? Or is it even generalizable to a larger population? Is it externally valid, internal valid? Concepts we'll study in one of our courses, inshallah. Relevant. Is it relevant to practice or not? So I'll just let you see this 5-6 minute video while I answer this question out from my hospital and try and see if you can learn something from this nice video which I found online on clinical trials.
Understanding clinical trials. Clinical trials are research studies that evaluate new ways to improve treatments and quality of life for people with diseases like pancreatic cancer. Clinical trials are an option for all patients to consider and play an important role in improving healthcare. Results from clinical trials help develop new drugs, new diagnostic tools, and new clinical procedures. Clinical trials can also spur new questions for research, which lead to new discoveries over time and help us to better understand diseases like pancreatic cancer. All the drugs and tools we have today were made possible because patients volunteered for clinical trials. Every clinical trial has what are called eligibility criteria. For example, a patient may need to have a certain medical condition or gender or other specifics to meet the criteria of participating in the clinical trial. Otherwise, they are not eligible to join that clinical trial and could seek another trial that better fits their situation. Patients who want to volunteer need to understand the potential benefits and risks of participation in a clinical trial. Patients may personally benefit from receiving new therapies or procedures. They receive close supervision from clinical staff. Sometimes the cost of the new drugs or testing is free or waived. Participants may also be part of a research breakthrough that helps thousands of future patients. Patients face the risk that they may not benefit from the new treatment. Additionally, participating in a clinical trial requires commitment. Trials can be lengthy or demanding and may require extra treatments, extra time in the clinic, or tests or costs that are not part of standard care. It is possible, if the study is randomized, that they may be part of the control group that does not receive the new drug being studied. Informed consent is a process that helps patients make an informed decision about whether to join a clinical trial. An informed consent document will be provided that explains the purpose, procedures, and potential benefits and risks of the trial. There will also be thorough discussions between the doctor and patient so that all questions can be clearly answered. In late-stage clinical trials, participants are generally split into two groups at the beginning of the study. One group receives the standard treatment plus the new treatment. This group is called the treatment group. The other group receives the standard treatment only and does not receive the new treatment. This group is known as the control group. A computer is used to randomly assign the patient to either the treatment group or the control group. Randomization helps to prevent bias because neither the patient nor the doctor can choose which treatment group a patient is assigned to. Some clinical trials may want to study the effectiveness of adding a new treatment to a standard treatment to see what works better. In these studies, half the patients will receive the standard treatment plus the new treatment, and the other half will receive the standard treatment plus a placebo. A placebo is a treatment that looks identical to the new treatment, but contains no active ingredient. These types of studies are often called blinded, meaning that one or more parties involved in the trial do not know which patients have been assigned to which treatment. Single-blinded means that only one party, either the researchers or the patients, knows which patients have been assigned to which treatment. Double-blinded means that both the researchers and the patients do not know which patients have been assigned to which treatment. The strongest, most reliable results come from clinical trials in which the treatment is double-blinded, the patients are randomized, and the study has a control group. This is called a double-blinded, randomized, controlled clinical trial. Clinical trials are carefully monitored to protect patients, and several layers of review committees are involved to protect patients from any possible harm. Patients can leave a clinical trial at any stage. However, they should discuss this with their doctor before exiting to ensure the safest procedures are followed. There are 4 different phases of clinical trials. Safety is emphasized in each phase. If a trial does not have a positive or safe outcome in phase 1, then it will not be allowed to proceed to a phase 2 trial, and so on. Before a phase 1 trial begins, there is a phase 0, where the new treatment has already been found to be safe in animals or healthy human volunteers. In phase 1 clinical trials, a new treatment is tested in a small group of participants for the first time to determine a safe dose and see how the treatment affects the body. Phase 2 is conducted with a larger patient group than the previous phase and studies how well a new treatment or combination of treatments works for a certain type of cancer, like pancreatic cancer. At this stage, the eligibility criteria become more restrictive. Phase 2 studies can be either randomized or non-randomized. Phase 3 trials are conducted in an even larger group of participants. This phase involves a randomized design, comparing the new treatment to the standard treatment. If a phase 3 clinical trial is positive, typically the new treatment will be approved and licensed for use in the United States by the Food and Drug Administration, or FDA. After the treatment has been approved, phase 4 clinical trials involve monitoring to continue to assess safety, effectiveness, and optimal use. Clinical trials play an important role in moving science forward and can be a hopeful option for many patients with pancreatic cancer. To learn more about clinical trials and how to participate in one, please talk to your doctor. You can also visit the National Pancreas Foundation's Clinical Trial Resource Center.
Okay, so with that, I just want to take you through a small, quick fire journey through a randomized controlled trial that I participated in with my team, including Dr. Atta out at National Guard hospitals in Jeddah, Saudi Arabia. And this happened because as part of accreditation of the organization, people were trying to get accredited from Joint Commission International, which certifies hospitals in different parts of the world. And of course, there are examples in Pakistan like Aga Khan and Shifa and Shaukat Khanam and Pakistan Kidney Liver Institute and others, which have also achieved accreditation by Joint Commission. And in that, one of the things that's recommended is to try and have multidisciplinary pathways for uh, different diseases that are common or frequent or high risk or error prone, because some evidence existed that some pathways work. So when they asked us to do that, we at the Department of Medicine and allied teams in pharmacy and nutrition and nursing and others, we got together and said, why don't we do this? But let's also look at whether some of these pathways, if we implement them, will actually work. So this exciting journey talked about. So we looked at what clinical pathways were, and we found that these are evidence-based, outcome-centered, and time-bound protocols and policies that are defined how a patient will be treated if they come into the hospital with a certain diagnosis. And one of the things that was important was that how could we provide patient-centered care to these people so that they actually feel they're the most important persons in the hospital getting their therapy. So not being subject to the hospital's processes, but hospital being, serving as a guest in the life of the patient. So, there was some preliminary evidence, like we said, that each time you do research, you have to see what has been done before. So you have to go back and search what has been done and what was the gap. So when we looked at, some evidence, it seems like there was some evidence of reduction in all-cause hospitalization and costs, as well as improved survival when you come together as a team and provide care like that. But you can also provide care which is protocolized or based on protocols, but there may be some variances, and one had to define that as well. But there was some evidence that if you put things together in a decision support system, computerized decision support system, you may also have certain benefits in some diseases like which are chronic illnesses. There was also some evidence that if you use in heart failure, for instance, computerized, order sets for uh, for certain diseases like heart failure, one could improve outcomes. So there was some evidence like that, and there are some audit tools that also exist. But if you look at the actual evidence, there was some evidence from surgical literature that if you replace somebody's knee, do total knee arthroplasty, then there was some decrease in length of stay and satisfactory clinical outcomes, and a majority of patients could be discharged home with outpatient physiotherapy and minimal complications. And maybe the same thing exists if you have pathways to provide care for pediatric age group people for asthma. Similarly, there were some unanswered questions that at the end of life, somebody is dying, putting in pathways, are they useful in providing comfort at the end of life or not? There was no real evidence. So some RCTs were obviously then needed to provide evidence in some of these other cases. Similarly, in acute coronary syndrome, not very clear that for heart attacks and acute coronary syndromes, pathways actually benefit or not, or usual care is as good. What about stroke? Is that something that would help in improving outcomes? In one study, which was not exactly randomized controlled trial, community-acquired pneumonia showed a favorable outcome. So in another study, there was no significant relation between implementing aspects of a pathway and improvement in outcome. What about heart failure? Well, there was an RCT, and they seem to be showing that pathways are effective in reducing mortality and unscheduled readmissions, at least for heart failure. But are they cost effective? For instance, in acute peptic ulcer bleeding, it seems like they're cost effective if you have a pathway and short stay in the hospital. But when you put all these evidence together as in the form of systematic review or meta-analysis, the Cochrane collaboration suggested that yes, there was some evidence that factors which favor pathway are a little better than those which favor usual care. But the rest of those evidence is very close to this midline, which is the point of no effect, either touching it or crossing it, touching it or crossing it, or very near to it. So the evidence was conflicting, but there was some signal that if you do pathways for multiple diseases, maybe there may be some benefit. So based on that, we thought we could still add some information to the literature by using our top 10 admitting diagnoses, maybe at least developing pathways for most of them, but at least using 5 or 6 of them in randomized controlled trials. So it was a challenge and an opportunity because there were paucity of randomized controlled trials which looked at pathway care versus usual care in real life settings. So we came up with this CHAMP-PATH studies, collaborative healthcare professionals approach in monitoring of patient-centered outcomes through pathways. And my co-investigator, Dr. Atar Rahman, is the chairman, Science International, and we had a very good period developing these pathways and taking the journey subsequently as well. And when we thought that let's see if this gap can be answered. And by doing that, we had to define whether our pathways would decrease the length of stay and they would improve patient related outcomes, and what would be the determinants of the length of stay in the hospital, and whether they would have what the effect of pathway care would also have on 30-day rehospitalization rates for the same diagnosis. So if somebody with pneumonia is discharged, do they come back with pneumonia again within a month? or not by implementing pathways or no pathways. So one of those was about acute kidney injury that we studied out of those 4 or 5, at least one of them which got published. And this clinical pathway would have a plan of care, but after admission, from with certain protocols, patients were randomized to either pathway or usual care, and then the information would go to everyone: nurse, pharmacist, social worker, patient educator, nutrition, all the way till discharge, and then one would follow the outcomes and see the length of stay. So our null hypothesis, which is always important for any research question, was there will be no difference between the length of stay between the usual care and the pathway care. Our alternate hypothesis, which we were hoping for, was really that pathway care would be different from usual care. This was a pragmatic that means mirroring real life RCT, parallel design, single blind. Patients did not know if they want pathways or not, but the teams who were providing care, obviously, it was difficult to blind them, so it was single blind study, and which is quite common in so-called pragmatic RCTs. The sample size for acute kidney injury was calculated at 38 patients with a power of 80%, and alpha type 1 error rate of 5%, and with the length of stay uh, you know estimated being 5 plus minus 1.8, and a reduction in length of stay of 2 days was the aim. And this calculation, all these things, we usually discuss them in detail in randomized control trials workshops that we'll be offering in October, inshallah. So the primary objective was, in terms of length of stay, was the pathway care superior to usual care, and we estimated it will be at least 2 days better, 2 days decrease in length of stay. And secondary objectives were in-hospital mortality, the determinants of the length of stay, 30-day readmission, and patient-centeredness as measured by a qualitative survey of the patients who were subjected to these pathways. And the on-call residents would determine the eligibility criteria, inclusion, exclusion, take an informed consent. Then the patient would be randomized and allocated to either usual care or pathway care, and would be admitted to any one of the clinical teaching units, and the referral pathway would be generated if they are on pathway care, or usual processes will continue, and the multidisciplinary healthcare team would get involved right from the beginning in the pathways, and then what would see the outcomes. And so the eligibility criteria in summary, if the creatinine increased more than 50% from baseline, or age was over 14, acute kidney injury was a major diagnosis, and if they were hemodynamically stable in terms of blood pressure and other vital signs. And we excluded people for pregnancy, ICU patients, stage 4, 5, chronic kidney disease, kidney transplants, obstructive kidney disease, and various forms of nephritis, because you either exclude people for confounding, which is another thing we'll study at some point, or safety, or feasibility. And then, the various forms of acute kidney injury were included in there. And this was the pathways that were put in the computer for all these different diagnoses for acute kidney injury. These were some of the things that were various protocols that were assigned to some people by chance alone, and the others used to get usual care. Usual care we provide to people with acute kidney injury. So, but this was all like pathways. So we don't have to have a new medicine or a device. We can have a behavioral change to study, or a management strategy change to study in pathways. So eventually 98 people were eligible, but a lot of them were not, they were assessed. A lot of them were not eligible because they did not meet the criteria for inclusion. 38 were randomized, out of which 18 went to pathway care, usual care, 20 went to that. Some were lost to follow-up, some discontinued pathway. But eventually, all those who were randomized were analyzed. So 18 were analyzed here, 20 were analyzed here, which is known as the intention to treat analysis, which also we study in our randomized control trials workshops. So these were baseline characteristics which were fairly evenly distributed by randomization. So if you have good randomization, most things become fairly evenly distributed, but there can be some mismatch. For instance, seems like for some reason, uh, more heavy patients went on pathway care as opposed to usual care. So you can't control everything that happens after randomization. But when we looked at the length of stay for the pathway and usual care, there was no difference. So pathways, in fact, they were 4.96 days. Usual care were 4.8 days of admission. But this is the intention to treat. So as randomized, as analyzed, which is a concept we study in our workshops. And this is the per protocol. These were the ones who actually received a pathway. So here, the ones who actually received the pathway were discharged in 3.12 days compared to those who were on usual care. So we had a 1.5 day decrease in length of stay on the per protocol analysis. All it suggests is that even though this p value here is not significant, there is something in the pathway that decreased the length of stay. And if we do larger studies, maybe in the future, one can show a difference in acute kidney injury. But there was in terms of mortality or people dying in the hospital or 30-day readmission, there were no patients who had that. We also looked at whether improving creatinine level and doing an ultrasound within 24 hours was predicted on the length of stay. It was not predictive of the length of stay in the pathway group. And patient satisfaction in the usual and pathway care was also pretty similar. No p value was not significant. So, technically speaking, one could say this was are pathways effective in acute kidney injury? No, there's no difference in pathway care versus usual care in terms of mortality or patient centeredness or 30-day rehospitalization. But per protocol analysis suggested some clinical benefit of doing this. So, see, if every research can give you a signal that can be useful. So, pitfalls, well, this is like a supportive care study. There could be contamination, which means that people who were getting usual care got aware about certain intervention in the pathway group also by talking to each other. These are medical residents, and maybe that's why the groups became very similar. And so in terms of pathway adherence, and there may have been a problem with variances, but it was also community acquired. We excluded hospital acquired and critically ill patients. Maybe that's the reason it didn't make any difference. So, but anyway, I just thought I will present this as an example, since myself and Dr. Atta were deeply involved in this particular study, and I thank all my co-investigators for participating in that. And with that, I just want to make a brief announcement that inshallah we will be conducting ABCs of randomized controlled trials for health professionals and students on these dates, more or less. Hopefully, we will stick to these dates or with minor variations if there are any. And these are coming up in the month of October. But I want to show you some sample questions from some of these workshops. So, for instance, one question is, usually a data safety monitoring board requires this analysis at selected points during a clinical trial to ensure patient safety or drug efficacy. If you know the answer, somebody can speak up. Or you can say, I don't know. You can unmute your mics now. And if somebody wants to answer question one, go ahead. Question two is also up for grabs: ability to make one's own decision without coercion. What is it? Is it autonomy, capacity, or I don't know? Question three: this technique ensures proportional representation in a trial. Is it stratified randomization? I don't know. Permuted block randomization. Question four: in this design, the effect of a new treatment is not worse than that of an active control by more than a specified margin. Is it superiority, non-inferiority? I don't know. So the answers are all there, and in our workshop we discussed the pros and cons of some of these responses. Another question is, 1 divided by absolute risk reduction gives this number, which reflects the minimum number of patients required to be treated to show benefit in one subject. Is it number needed to treat? I don't know. If efficacy number. Now, question 23. This margin determines how comfortable you are that the sample mean and population mean both lie within this margin. Is it confidence interval? I don't know, or standard error of the mean. So once we do these workshops, we will realize that most of these things we'll be able to answer ourselves, and we'll be able to participate in a clinical trial, because most of us health professionals, if we are health professionals, work in centers which are like hospitals where most people have nursing, most people have pharmacy, most people have radiology, most people have an area like patients who can be assessed in different areas who are enrolled after informed consent. Most people would have an ethics committee in the hospital or center which will review research protocols and make sure things are safe for the volunteers who are participating. Most people will have IT, data management center, and a lab. And all one needs to do is have a proper area for storage of investigational medicinal drug. And you have your clinical trial unit, which can be inspected and approved by following some guidelines, which are based on DRAP requirements, at least in Pakistan or other countries, similar requirements, and one can participate in clinical trials, because our legacy starts not just from James Lind, who first did randomized controlled trials by subjecting sailors to either lemon juice or no lemon juice and seeing whether it develops scurvy or not. We celebrate May 28 or May 20 for him as the International Clinical Trials Day. But the first to utilize concept of control groups was Al-Razi, 1,000 years ago or more. And then again, 1,000 years ago or more, Ibn Sina described the 7 rules of drug experimentation for new drug development, which is pretty much the same even now. So these are heroes that we can always emulate. And thank you and best wishes for an exciting journey through translational research. I thank Dr. Atta and team and Science International for giving me the opportunity. And I end my presentation here, and I'm happy to take any questions if there are any, either in chat or you can unmute for that. Thank you very much. Any questions? Comments? Okay, so there are certain so meta-analysis, systematic review, Aisha Masood Saiba, you are asking, and I think the areas also already answered. They're right on the top. But evidence pyramid in terms of clinical research, yes. But if you're looking at the EBM pyramid that is based on systems and synopsis and synthesis and all that, even collection of meta-analysis and all, usse bhi upar is that your clinical decision support system. But yes, in general, when you're looking at expert opinion and cross-sectional and case control and cohort and RCT, then above that are the systematic reviews and meta-analysis. Okay. Any other questions or comments? Please unmute yourselves. And if there's any question, I'm happy to answer over the next couple of minutes. Do you have Dr. Atta still online? Can everyone unmute themselves? And if you have any questions or comments on today's session, whether you found it anything that can be an exciting area to get into it.
Thank you so much, Dr. Sam, for this informative sessions. I joined late, but it was really informative for me. I'm Dr. Tosi Iqbal from Punjab University.
Thank you. Thank you. God bless you. Anyone else who would like to make any comments or have any questions related to the presentation or the upcoming workshops that we are going to be doing in October? So
Thank you, sir. We are eager to learn from you in the next following sessions.
inshallah, I'm looking forward to interacting, and please share the information widely so that we can have a good learning community and an interactive. So we're going to have 3 sessions, inshallah, 2 hours each in on RCTs. I believe there's going to be around the 10th, Friday the 10th, 17th, and then Wednesday the 22nd. And the first 2 sessions will be on different aspects of the important concepts which are important for any clinical research, and how to formulate a research question and eligibility criteria and confounding and all those things, and related question answers. Second session will be on the specifics of the randomization and allocation concealment and blinding and intention to treat analysis, followed by a quiz. And the third session will be on all the rules that govern the game, the ethics of it all, the informed consent, the good clinical practice, the ethical disasters that we have to avoid based on historical wrongdoing. So those sessions will be coming up in October. Watch out for the science announcements for that, and hopefully, inshallah, we'll have a good learning cohort, and we'll all learn together. So thank you very much for again Science International, Dr. Atta team for joining today. And this session is available as a recorded session, and we will try and share that with you subsequently very soon. Thank you. Assalamu alaikum.
Thank you so much, sir.
Thank you.